mention real quick. So Friday's exam will be the same format that we had for our first exam. So there will be 25 multiple choice. There will be a short answer where you get 4 possible topics and you go ahead and you pick 3 out of the 4. And then 1 long question where you get 2 topics and you get to choose which of the 2 topics you want. And so it'll all be done in La Lima. So it will be set up as a test in La Lima. So you guys would log in, first log in to Zoom, just like you've done today, so that I can see everybody's present. And then the TAs and I can actually see all of you working on it. So please have your video enabled for that. So if you're concerned about having that be a big distraction on the screen, of course, I think on your screen you can always minimize and not have the video showing. And then the actual test itself will be open for you to take in La Lima. And so that you'd open up your web browser, go to La Lima, and proceed to click begin on the test. And then once you click begin, you'll have 50 minutes to complete the test. And I think that should go very well because we sort of tried that out last time and it seemed to work fine. In terms of how the questions will be presented to you, you'll be able to move back and forth between all the multiple choice questions so you can answer them in whatever order you want. And then the short answer part will be its own part of the exam, part 2. And then the long answer will be part 3. And I believe it can be set up so that you can actually move between the different parts freely. But I will go ahead and set that up, and then I will send you guys an announcement to kind of go over that. Okay, so for now, let's go ahead and review the material that's going to be on the test. So we were talking about this idea that mutations happen at random in our genome. And a given mutation, you can think of it as maybe either hindering or potentially enhancing the function of a protein that is affected by that mutation. So really then, the mutations that matter and the mutations that we end up seeing in something like a cancerous tumor, there's a bias there, right? So we've actually selected for the mutations that actually matter. Right? But then that's easy to forget then that in reality, other mutations are happening all the time. But if those other mutations don't really matter, they're not selected for. So we looked at this idea that in tumor suppressor genes, you might have mutations that hinder the function of a tumor suppressor gene. And so once the cell loses that tumor suppressor gene's function, that might favor transformation. Likewise, mutations in oncogenes, the mutations that would matter would be the ones that actually enhance the function of a tumor suppressor gene. So when we looked at RAS, we appreciated the fact that when you look at the whole RAS protein, the majority of the mutations that you see in tumors that involve RAS all have a point mutation at around amino acid 12 or 13. So what I want you to think about here is that because mutations are random, there's equal probability that you're going to have mutations that actually end up enhancing the function of a tumor suppressor gene, or the opposite would be a mutation that actually hinders or prevents a proto-oncogene from functioning. But why don't we see them? Why don't they matter? And what I wanted you to think about was the fact that, well, if you actually had a mutation that enhanced the function of a tumor suppressor gene, for example, made P53 or RB overactive, then that might result in that cell just no longer being able to divide at all. So it doesn't matter because that cell just goes away. Likewise, if you have a mutation that hinders the function of a proto-oncogene, same thing. You're actually just slowing down then the ability of the cell to go through the cycle. So you just wouldn't see it as part of the tumors that as part of the mutations that you see in tumors. Remember the analogy that I gave you having to do with this idea of bias. The fact that we're looking at tumors means that we are selecting for just seeing the mutations that mattered. Right. So I said, if you go to a big mall like Ala Moana and you randomly surveyed 100 people and asked how many of them took the MCAT, you know, you might find like 3 or 5% of them did. But if you actually go and do the same survey of 100 medical students, you would find that 100% of them took the MCAT. Right? And so that doesn't mean that people don't take other tests as well, but because you're biasing yourself by just looking at medical students, you see the MCAT as the thing that stands out. Loss of heterozygosity is a very important concept in cancer development. We said that for many tumor suppressor genes, we have one copy of, let's say, RB from mom, one copy of RB from dad. If they're both wild type, you're fine. Right? If one of them gets a hit, either through random mutation or you inherited a genetic variation, then you are heterozygous for it. But that's still okay because you have another good wild type copy to be able to produce the normal RB protein. So the problem happens when you lose that heterozygotic state and you actually then lose both copies. So that's why loss of heterozygosity is such a big deal. This was the data to remind you that if you are looking at something like familial retinoblastoma, individuals who are born with already being in a heterozygous state because they inherited one bad copy of RB, they will develop bilateral tumors in both eyes. Right? Those who spontaneously get a hit in their RB, they most likely would only have a tumor in one eye, not the other. If you then follow over the lifetime people who had bilateral tumors versus people who had just tumors in one eye, and you just looked at the relative probability of them developing tumors in other parts of their body, what we see is that the patients who had familial retinoblastoma, right, have a far higher risk of developing tumors elsewhere in their body than those who only had a somatic mutation in RB in one of the cells in their eye. Right? So that really highlights this idea of loss of heterozygosity and how that can influence the development of cancer. We discussed the cell cycle. So remember that the different phases of the cell cycle are encouraged or regulated by waves of cyclin production. And we said that cyclins, such as cyclin D, E, A, and B, can be thought of as just simple little adapter proteins that help reshape the cyclin-dependent kinase partners that they bind to. And the CDKs are the kinases. So these are the proteins that actually know how to transfer phosphate groups onto a substrate target, like another protein. And that would then lead to the activation of a particular series of proteins or a particular pathway in the cell. But without the cyclin binding to it, the shape of the CDK is not correct in order to interact with different partners. So we said that we can think of the cyclins as being, for example, the different types of bit attachments here in a screwdriver set, and the CDK being the actual part that does the work. So in this case, the handle of the driver. Right? So depending upon the combination of the two, that determines what kinds of targets you are acting on. So that's why the very elegant and seemingly simple act of having different waves of cyclins, like cyclin D, go up during G1, or cyclin E or A go up as you get into S phase, that can end up activating a whole bunch of different pathways because it allows the CDKs to talk preferentially to different proteins, right? So it's not just now activating one or two pathways, but entire programs that are associated with the cell functioning properly in G1 versus S phase versus mitosis. Importantly, we talked about this idea that differentiated cells want to stay in G1, because that's when they're receptive to different types of signals instructing them what to do. And it's during G1 when they carry out their normal fully differentiated cell functions. Part of this, if they are continuously receiving mitogenic signals, you would get an increase in cyclin D levels. So you would have more cyclin D partnering with CDK4. So that begins to affect the phosphorylation status of RB. So here the dark green is RB, and remember RB functions to guard the progression of the cell cycle because it binds to and prevents E2F transcription factors from turning on certain genes. So as you get more and more cyclin D in G1, then you would actually have as a target of mitogenic signals, right, the production of more and more cyclin E, and that when paired with CDK2 results in increased phosphorylation of RB, and in the hyperphosphorylated state, RB then can no longer effectively inhibit the E2F transcription factors. So we said that once you start building enough of the cyclin E CDK2 complexes, you are now crossing the R point. Right? And the R point is that point that past which the cell cannot back up and decide to just stay in G1. It's starting then to allow the E2F transcription factors to go to work turning on the genes that make proteins that are needed for S phase. Part of this is this feedback loop. We said that the more E2F activity you have, E2F itself controls its own gene, so you get more and more E2F signaling, and cyclin E is another target of E2F, so you get more and more of this loop. And because of that feedback, it's very hard to imagine that the cell could somehow back out of this. So that's what we mean when we say the R point really is a commitment point or a restriction point, that the cell has to get through the rest of mitosis, and whether or not it encounters problems or not, right, it's committed to at least try to get through mitosis. So as a reminder, all of these pathways that are pro-proliferative and anti-proliferative are all happening at the same time in the cell. So we said that the cell cycle is really a two-footed driver, one foot on the accelerator, one foot on the brakes, both being pressed down, and it's just a matter of which one is being pressed harder that ends up nudging the cell one direction or the other. This is a nice figure. We talked about it a lot, but remember in this figure here, the orange pathways are accelerating factors. The blue ones are braking factors. So in a complicated figure like this, it's easy to, or it's better to start by looking at a target. For example, here's the expression of cyclin D1 protein from the cyclin D1 gene. It's stimulated by these three different pathways here, right? So the arrows mean that these are stimulatory pathways. If we follow this one, we see, oh, yeah, beta-catenin activity increases cyclin D1. But normally, beta-catenin, if we work backwards now, is inhibited by APC. So APC helps form that complex that causes beta-catenin to be degraded. So that's why APC's normal effect is inhibitory. But you go above APC and you say, if Wnt signals are arriving on the outside of the cell and are received by, for example, the frizzled receptor, that inhibits the ABC or the APC. So it's a double negative. Inhibition of APC relieves the inhibition of beta-catenin. All right, so that's how we read these types of flowcharts. We see here several different oncogenes that we've talked about, like RAS, for example, and MYC is in here as well. And then we see some tumor suppressor genes that we talked about, PTEN, APC, retinoblastoma. Right? Really, the center part of this figure is the fact that all of this kind of converges on RB, because RB, again, is controlling the R point transition. This is also a nice reminder to go back to your notes and review what these different CDK modulators are, like P15, P16, P27, right? And just remember how that they play an important role in sort of subtly controlling whether the cell cycle moves forward. Talking about Wnt and beta-catenin, we looked at this wonderful example here of the colonic crypts and how the epithelial cells in the crypts of the intestinal lining are encouraged to proliferate and differentiate. We said that stem cells, one of the ways that stem cells are protected from mutations is they are relatively deep in the tissue. So they are at the bottom of the crypts here. So the gray things are the few stem cells that you have, maybe 1 in 100, maybe 1 in 500 of the cells that you'd find would be a stem cell. Down here you have the stroma, right? That's the connective tissue that's below the basement membrane of these epithelial cells. And the stroma is sending Wnt signaling, right? So the little red arrows. So the stem cells receive that encouragement, they divide. So stem cells are all about proliferation. And so they make these transit amplifying cells. These would be the daughter cells that then proliferate, because again, they're still receiving beta-catenin signals in the form of Wnt. Right? So during this area down here, because you keep getting Wnt signaling that's turning on beta-catenin, you have MYC and MAX working together. And remember, this is a really nice image over here that shows the staining in green of these cells are ones that are proliferating, and MYC and MAX is working that way. As those cells move farther and farther away from where you have the Wnt signaling, they lose the beta-catenin signaling. So beta-catenin starts switching off. And as a result, MAX and mixed work together and start controlling differentiation. Right? So then the cells are now less concerned about proliferating. They want to differentiate until they achieve their final fully differentiated form, like this one here. Right? And the cell might stick around for a few days before it's physically lost, shed into the intestine, the lumen of your intestine. That's why we said these cells up here are very exposed to mutagens. So up here, if you have all the chemicals and things in the food that you're eating, of course, it's going to be these cells that take a bigger hit in different mutations, but it's okay, because if this cell gets mutated, it's just going to be lost in a few days. Whereas the stem cells are buried deeper, so they're protected. And again, over here, you can see it has now shifted. The cells no longer show the green marker for proliferation, they show this red marker for differentiation. On the right part of this slide was giving us an idea of what would happen then if we had a mutation that messes up the proper beta-catenin signaling. So for example, a mutation that disables APC, or a mutation that enhances beta-catenin function, right? That might lead to then these cells up here always just wanting to differentiate, always wanting to proliferate, and you might have the start of a tumor. So the cell cycle is guarded by p53. Right? So the genome is closely monitored by p53. And we said that p53 is probably the most important tumor suppressor gene because it has to make decisions about when the cell is undergoing damage to its genome and there are defects that happen, either mutations or breakage in the chromosomes. p53 has to stop the cell cycle. So this allows then some time for DNA repair to happen. And if the DNA is then repaired, then p53 stops getting the signal that there's damage, and then p53 shuts off again. Right? If you continuously get the signal that DNA is damaged, maybe p53 will cause the cell to go into senescence. Or if things are really bad and you have very strong p53 signaling, in other words, the nature of the damage is so great that you get a ton of p53 activated and nothing changes, then maybe p53 would initiate apoptosis because the cell cannot fix the problem. So go back and review how p53 functions, how it's regulated by factors such as MDM2, so that you have a good understanding of how it is that our cells control p53 activity. An important thing that we started discussing was this idea that there has to be limits on cell division and the limits on cell lineages. So when we think about early totipotent cells, like in the early embryo, that still have the ability to become any cell in our body, as they differentiated through time, and as we grew and developed, then your body was maybe had a lot of these pluripotent cells that still had the ability to be many kinds of tissues. But these days in our adult bodies, what we have are multipotent stem cells. Right? So these are far enough down the differentiation path that they really can't back up and become some completely other type of cell, but they can become any number of cells in a small group. So for example, the hematopoietic stem cells in our bone marrow that can become any of the types of cells that make up our blood. Right? But that stem cell doesn't know how to become something like a nerve or a muscle or a gland. So these stem cells persist, right? And they give rise to fully differentiated cells that maybe proliferate a few more times before they undergo apoptosis. When cells are pushed too hard to divide, they lead to senescence. And we said senescence is when the cell looks like it's going into G0, but it's different than G0 because G0 is just taking a break from the cell cycle. And when things get better, it can always go back into the cell cycle. So for example, if there was a depletion of nutrients or other types of things, the cell just needed to take a break. Once things get restored and you have nutrients again, the cell can return, if that's just G0. But senescence is a special type of G0 where the cell cannot return. Right? And that often happens because of things like crisis, when the telomeres get too short. So actively dividing cells like these fibroblasts over here might have this appearance. Once they enter senescence because of too many cell divisions, their dividing rate of division begins to plateau. They took on this kind of weird shape. It's kind of like a fried egg almost appearance, which is very typical of senesced cells. And in this state, the mitogenic pathways inside of the cells are all deactivated. So even if you gave these cells a lot of growth factor, they're not going to come back. So they're not dead, but they're just not going to do anything anymore. P53 and telomeres are very closely related in terms of how they're working together. We said that if you lose P53 and the cells are dividing, the telomeres are getting shorter and shorter and shorter. So normally, if this were a fully differentiated cell, after a few divisions, these red telomeres would be too short and the cell would be hitting crisis and might undergo, it might be senesce and not move forward anymore because P53 detects that. But if you don't have active P53, the telomeres will get too short and then you run into problems because then you risk losing the actual important genetic information that's in the main part of the chromosome. But also, if you lose the ends of these chromosomes because there's no more telomeres, they might stick together. And then when the cell divides, these chromosomes get torn apart and you get these weird hodgepodge mismatch chromosomes, which are very clearly deleterious to the proper functioning of the cell. Right? So this period of genetic instability then might be sort of on this figure here, this middle part, where the cells of a transforming founder of a tumor are undergoing these breakage, fusion, bridge cycles, leading to accelerated mutations for getting more and more messed up. But the important thing here is this can only go on for so long, because if this keeps going through these BFB cycles, the genome will be so messed up that the tumor cell just can't even survive as a living cell anymore. Right? So in order for this to keep going, or the cell to keep surviving and actually become a fully malignant cancerous tumor cell, this BFB cycle has to stop. And that happens if one of the mutations that by chance are stumbled upon by the cell is the reawakening of TERT, telomerase, right? Because then the telomeres start extending again, and then you stop going through this crazy, unstable breakage, fusion, bridge cycling, and whatever messed up genome the cell ended up with, right, that's still good enough for it to keep living, but clearly is all messed up, that then poses a real problem, because that then is your cancer phenotype. right? And this continues to mutate now, maybe through point mutations or other things. So that's why we said in order for cancer cells to gain immortality, two things have to happen. You have to lose tumor suppressor gene function. So for example, in this case, we're saying you might imagine that certain tumor viruses that express tumorigenic viral proteins like SV40 large T antigen that deactivates P53 and deactivates RB. Remember, SV40 large T antigen can actually bind to RB and prevent RB from properly binding to the E2F transcription factors. So RB can no longer control the cell cycle there. And remember, SV40 also binds to P53 and prevents P53 from being able to go into the nucleus and turn on genes. So that's one step. So that'll lead to the breakage fusion bridge cycle, where suddenly the genomes get progressively more and more messed up as chromosomes are torn apart and then improperly joined together. But the second part has to be that at some point, the transforming cells reactivate the expression of TERT so that you end up getting the extension of the telomeres to put an end to the crazy BFB cycle. And then that cell actually, unfortunately for us, is able to continue surviving in a very messed up cancerous form.